So what about mistakes? Because this is a lot of copying. And if you've ever had to make, say, if you had to go to the photocopier and you had to make 100 copies of a 100-page book, you can pretty much bet that you're likely to get a few mistakes. So does that happen in the reproduction or the replication of DNA? And the answer is, of course it does. Anytime you're kind of making copies of copies over and over again, you're bound to have some mistakes made. And the same thing occurs in DNA replication. So DNA polymerases actually do a really good job. They kind of proofread the newly made DNA as they are creating it. So as they lay down the next nucleotide, they kind of check it against the template. And if it doesn't match, they will kind of immediately fix that. But sometimes those errors get through. In humans, there's about 130 known DNA repair enzymes. So there are enzymes, which are, again, remember, are proteins, that their specific job is to check and double check the DNA and make repairs if they have to. So you can have problems if you have non-working DNA repair enzymes, because you have DNA damage and mutations that build up over time, and that can potentially lead to cancer and other problems. So you have a lot of copying of genetic information going on inside your body, and you basically need a lot of proofreaders to double check and make sure these copies are correct. And that's what the DNA repair enzymes do. So the DNA polymerases do a really good job of catching problems right off the bat, but if they don't catch those problems, there are other enzymes that catch them a little bit later. So in mismatch repair of DNA, you have repair enzymes that come in and correct errors in base pairing. So if you have an A with a C when it's supposed to be with a T or something like that, you can have enzymes that come in and kind of fix that after the fact. And that process can be disrupted or made more problematic by various environmental factors. Like if you are exposed to various chemicals or radiation, cigarette smoke, and X-rays, those are all things that can damage your DNA and cause these repair problems that need to be done. And then also there are spontaneous changes. Even if you never were exposed to X-rays or radiation or things like that, the DNA spontaneously does have some mutations and damage that occurs also. So you need these various enzymes that are constantly proofreading and repairing for these problems. An example we're going to take a look at is called nucleotide excision repair. And this is where a particular enzyme called a nuclease, actually cuts out and removes damaged sections of DNA, and then a DNA polymerase will come in and repair that or make a new section to kind of keep that portion of the DNA proper and correct. So here's an illustration from your textbook of a nucleotide excision repair in skin cell DNA. Skin cells are constantly bombarded with radiation from the sun, ultraviolet radiation. If you've ever been to the beach and gotten a sunburn, that's radiation damage from the radiation of the sun. So there's really no way of protecting your skin cells 24 hours a day, 7 days a week for all of your life. So this radiation damage can cause problems with your DNA. A good example is right here in this picture. If you look at the DNA molecule and you see this kind of buckled portion right here, the covalent linkage between adjacent thymine bases, see where that little connection is, and it causes them to kind of pull together and buckle? Well, that little linkage is a problem. It's not supposed to be there, and that's caused by ultraviolet radiation, and it causes this little buckling, which then causes an issue when the DNA goes to be replicated. So what happens is you have a particular enzyme called a nuclease that comes in, notes this damage, and basically cuts out or removes that section of DNA. And then you have a different enzyme, DNA polymerase, that comes in and repairs that and replaces that damaged section with the appropriate DNA molecules. And then you have DNA ligase that comes in and ties that together and makes it one continuous piece again. So this is a good example of the repair work that goes on inside your cells. This one's called nucleotide excision repair. And you have, again, about 100, 120, 130 different enzymes whose sole job it is to check and make sure that the DNA is lined out the way it should be, and then to correct any possible problems or mutations that have occurred. So do any of these mutations ever get through? And of course the answer is yes, they do. So let's talk a little bit about the evolutionary significance of mutated DNA or altered DNA nucleotides. Remember when I say the word mutation, almost everyone thinks of the word mutation as inherently bad or inherently deadly. That's not usually the case. Most mutations are neutral. They don't harm and they don't help the creature. So your body accumulates numerous mutations throughout your lifetime inside the DNA of your cells, but most of those mutations don't really have an effect on you. So not every mutation is harmful, and not every mutation can be helpful. Some of them tend to be, well, I would say of those that do create a change, most of the time that's harmful. So most mutations that cause a change tend to be harmful, but not all of them. Some of them can be beneficial. The error rate after proofreading is pretty darn low, but it's not zero. Your body does a great job of correcting problems with your DNA, but it can't catch every particular problem. Now, if these changes or mutations become permanent and they get passed on to the next generation, that can have an effect on the offspring. Now remember, if you have a mutation in a skin cell, that cannot be passed on through your sperm or your eggs. The only mutations in genetics that get passed on are the ones that would occur in the cells that create gametes. So if you have a mutation that affects your ovaries or testes that create your gametes, well then certain mutations can be passed on in the sperm and egg to the next offspring. These changes, these mutations that get passed on, they are the source of genetic variety. And that's what natural selection works off of. So when you look around the classroom and you see your 30 classmates and they all look slightly different, well, they look slightly different because they have different genetic makeups. And originally, those genetic changes and differences are caused by minor mutations. So while some mutations are harmful, some can be helpful. And if those mutations occur in the genetics of the gametes, then that can be passed on to the offspring. So if you had, let's say for example, that blue eyes is a mutation. Well, if that mutation occurs in the gamete production sites of the mom or dad, well, that blue eye can be passed on to the offspring. And then, or let's say it's a genetic mutation in skin cells. So then when that skin cell mutation gets passed on to the offspring, when those cells begin to go through mitosis, every skin cell will have that same mutation. So if it's a mutation that causes a particular coloration of skin, well then those daughter skin cells, when they divide and go through mitosis, every skin cell will have that same mutation because it's going to share the same genetics. So the evolutionary significance of this is this mutation process, these copying errors, are not always bad. Sometimes they are, but sometimes they can be beneficial. At any rate, this variety, these changes, that's what evolution and natural selection work off of to create all these differences we see in the life around us. All right, before we take a break, let's talk about one last problem here. It seems like you would think that this DNA process would be fairly simple, but it is fairly complex and it can be problematic. So let's talk about one last little problem with DNA polymerase. So we already talked about the fact that DNA polymerase has a couple of issues. It can't start a brand new chain on its own. It can only work in one direction. Well, let's talk about one last little problem here. In eukaryotic cells like ours, the chromosomes are linear. In other words, they're not circular like, say, in the bacteria cells. Our chromosomes are made up of long strands of DNA that are linear. Well, this creates a problem because the replication machinery that creates the new strands has no way of finishing off the 5' ends. And so we end up with unfinished ends on one side of our DNA molecule. And you do this over and over again, and you end up with kind of like unfinished ragged edges of the DNA molecule, of the chromosome. Well, this produces shorter and shorter DNA molecules with uneven ends and can eventually lead to losing genetic information. Now, this is only for eukaryotic cells that have linear DNA. The prokaryotic, the E. coli bacteria, and those creatures that have the circular DNA, they don't have this problem. But let's take a look at this problem about the replicating the ends of the DNA molecule in eukaryotic cells. So in this illustration, we're talking about the lagging strand of DNA here, the one that has to kind of work backwards. And so you see we have the next to last fragment here where we have the RNA primer and then the section of DNA, and then an RNA primer and a section of DNA. Well, then when we remove those RNA primers and we tie that together, we're left with this little piece that can't be finished because we have no more out here to build another RNA primer and then move back to the right. So we end up with kind of a jagged edge here on this one new strand. And then as that process repeats or continues over again, we end up getting shorter and shorter and shorter. So we end up with edges of the DNA that don't match, and we could eventually begin to lose genetic information because of this problem. So how does the eukaryotic chromosome, the eukaryotic DNA molecule, get around this particular issue? Well, it's an interesting problem solving that has come about. The eukaryotic chromosome DNA molecule has a special nucleotide sequence on the end called telomeres. Telomeres don't keep this problem from happening, but they kind of act as a buffer, a safety zone, so that we have kind of a bumper of information on the ends before you get to the real genetic information. So it doesn't cause this problem from happening, but it postpones the possibility of losing true, actual, important genetic information that might be near the ends of the DNA molecules. So the telomeres, kind of think of them as protective bumpers of nucleotides, and they are at the ends of the DNA strand. They're not actually genes. They don't code for a particular trait. They're repetitive strands of one short nucleotide sequence. In humans, it's a repetitive strand of TTAGGG. So it's 3 nucleotides, and it doesn't mean anything. It's not a gene. It doesn't tell the cell to make particular proteins, but it's just a repeating sequence of TTAGGG. Now the problem is we lose these telomeres with every single replication. So imagine you have important information on a long strip of paper, and every time you make a copy of it, you're going to chop off the ends of the paper. Well, if you want that important information to last as long as possible, you're going to add extraneous, extra information to the outside edges, so that when you chop off pieces, you're just chopping off the extra stuff that doesn't mean anything. And you can do that multiple times before you get to the important information. That's what a telomere is. A telomere is a section of the chromosome on the ends. It's just a repetitive nucleotide sequence. It doesn't code for anything, but we lose sections of that telomere with each replication. And that's all because of this whole end process on the lagging strand, where we have to have an RNA primer and then work backwards, and we get to the end and we're stuck and we can't finish that last portion. So the telomere kind of gets lost or chopped off. That's okay, because the telomere doesn't actually contain useful information, but every time this happens, we're getting closer and closer to the actual true genes that we could lose. So telomeres don't prevent the problem, but they postpone the possibility of losing genetic information. It's been proposed that the shortening of the telomeres is connected to aging. There's a whole lot of study going on that's really fascinating about what does that have to do with our bodies getting older and breaking down as we age. And a lot of that scientists think has to do with these telomeres. The shorter the telomeres get, that starts to create various problems that might be related to aging and eventual death. Here's a great picture, microscope picture, where they have stained the telomeres in orange. So this is the chromosomes of a mouse. And you can see at the tips of the chromosomes, there are these areas that they have stained and highlighted. Those are the telomeres. the repeating sequence that is there just to protect the actual genetic information further on down the line. So what about our reproductive cells, the gametes? Remember those germ cells that make the sperm and the eggs? If every time they divided and our DNA in those cells replicated and the chromosomes were shorter and shorter with every replication, well, eventually you'd have a problem. You would start to have gametes that were missing genetic information. Well, our bodies have a particular enzyme that's at work in the germ cells that produce the gametes called telomerase or telomerase. Telomerase. It's an enzyme that catalyzes the lengthening of the telomeres. So this is an enzyme that adds on to the end of the telomeres and makes them longer again. It restores the telomeres to their original length. This telomerase, man, that's hard to say, telomerase, is not active in most somatic cells. So in your normal body cells, this doesn't work. The telomeres get shorter with every replication. But in the germ cells that create the gametes, there is an enzyme that keeps this from happening so that every sperm and every egg has full-length telomeres on the end of the DNA, so the new embryo can start fresh with that process. A lot of people and a lot of scientists, sometimes you'll even see like late-night television telemarketers, they talk about somehow, what if we could kick on or make active this telomerase inside our somatic cells? Perhaps we could live for 200 years, or we could live for 300 years. Would our bodies age and break down if we could keep these telomeres full length and not be losing the ends of these telomeres with every subdivision of our DNA? That's an interesting question. There's a lot of study going on in that particular area right now. Let's kind of tie back this telomere activity to something else we've talked about. We know that cancer is basically cell reproduction out of control, where cells are reproducing kind of wildly and they're not behaving in the manner that they should. Well, you would think that because cancer cells tend to reproduce very, very quickly and very often, that their telomeres would shorten to the point where they would eventually kill themselves off. Well, what's very interesting is that in some cancer cells, there is evidence that the cancer cell has somehow turned on this telomerase enzyme that keeps that from happening. So you would think that in cancer cells, the body would limit the growth of them because the telomeres would eventually run out. Older cells that have reproduced very often inside your body, they tend to start to die and go through apoptosis, because when the telomeres get too short, it triggers the whole apoptosis process. But cancer cells somehow get around that, or some cancer cells that have been studied somehow get around that by kicking on this enzyme that we talked about, this telomerase, which is normally only active in the germ cells that create the gametes, but somehow the cancer cells kind of kick this on, and so they get around this problem of the shortened telomeres. And so that's another interesting area of study with cancer and telomeres and what cancer cells do to survive, even when normally the body would have them killed off or go through apoptosis. All right, we're going to pause right there. We only have one very short section left, but we're going to go ahead and take a break, come back, and we'll finish up by talking about the actual structure of the DNA, what it kind of looks like underneath a very high-powered microscope, and then we'll be done. So go take a break, come back. I'll see you in just a moment.